So when we speak about in vivo imaging, we can divide it briefly in neuroanatomical imaging, such as magnetic resonance imaging, computer tomography, or PET, or we can use optical imaging. So, and today I will talk mostly about the optical imaging. We can use two different methods, in vivo fluorescence, in vivo bioluminescence, and I will slightly brush both of them, but mostly I will talk about in vivo bioluminescence. So, what is the advantage of using in vivo imaging in CNS pathology? We can perform in vivo pathology in different disease models in mouse or in rat. We can perform longitudinal therapeutic studies, and I will give you a few examples how does it work really, and how can we really measure the output of our pharmacological intervention. And what is mostly important is that using this method can bridge the gap between basic and translational research. What we develop in our lab is basically the concept of two black boxes, the black box concept, and we can apply it to acute and chronic brain pathologies. Why is it important? For example, if you talk about the chronic neurological disorders such as Alzheimer's, dementia, or ALS, we know that these are the late onset diseases, and we know that we have a period of a silent pathology that can last for over 40 years, several decades. You live normally, but we know that there are early biomarkers, early and subtle signs of disease that we are still not able to detect by classical means. Contrary to that, in stroke, after stroke or traumatic brain injury, the people live normally up to 50, 60 years, then the incidence of the stroke increases, and after that there is also, if you survive a stroke, there is a silent evolution of the secondary injury. And what is really, really important, we know that traumatic brain injury as well as stroke are the great risk factor for developing dementias later on in life. In my laboratory, we developed a following strategy. We wanted really to visualize in 2 and 3 dimensions what is going on in the brain of ischemic animals. We developed several of mouse models in which we can visualize different elements of this ischemic injury. First of all, we developed a model for measuring astroglial activation, microglial activation, and these are the 2 elements of neuroinflammatory response. And we also developed a mouse model in which we can really follow neurological damage in a stroke. So the model that we use is basically, and this is the part of GlowBrain, is in vivo bioluminescence imaging. So our mouse model with express luciferase, this is the firefly luciferase under control of certain gene. And for example, once your gene that is involved in your brain in inflammation is activated, and when we inject the luciferin with this substrate, we will have a bioluminescence activation. The light will scatter through the brain, and we can image and visualize this signal from the brain of light animal. And I'm really happy that very soon this will be possible to do in Zagreb. This is, for example, this is the mouse that had a stroke, and we can see now the neuroinflammatory reaction in this mouse, and this is the live animal. This is just slightly anesthetized mouse, and we can follow it over time. So how does it look? So first, you have a mouse. We induced experimental stroke, and what is really interesting in this model, depending on the type of occlusion, time of the occlusion, we can modify the level of the damage. Our first question is whether this corresponds really what we were used by classical methods, whether this is really quantifiable signal. And when we performed classical immunohistochemistry, and this is activation of the same cells that we measured in vivo, we can see that we can really obtain the very similar ratio of the activation. And this is now really, really important. Now we know that quantifying this signal, it's possible in vivo. Now we don't have to touch the animal, but we can follow what is going on in vivo. So after that, we also, because the microglia are the other cells that we are particularly interested in, and these are the innate immune cells of the brain, like brain macrophages, we were interested to visualize microglial activation. And for that, we generated our own model in which we expressed luciferase and GFP, two different reporter genes under control of toll-like receptor 2 promoter. So if we go back, this is the schematic presentation. When we activate this receptor, what it goes, it finally activates NF-kappa B, which is a major inflammatory factor, to the molecule MYD88. So we wanted to see whether this is really what's going on in the brain, because this is our hypothesis. So you can see here the message RNA in the brain. We can see that the RNA message from this receptor is strongly activated. And if we remove part of the microglial cells in our different transgenic model, we can see that we can eliminate part of the signal. And using the GFP as a reporter gene, we can see here we can go from the whole animal to the cell, and we can go back into a cell and see exactly which cell type is affected, and it was the microglial cells. And this slide, you can see a single animal that we follow for three months. So what we learned? We learned two things. First, that inflammation after stroke, at least microglial activation and activation of innate immune response, is a chronic response. It can last for several months after. And we also learned that there are the signals that we can detect at the other parts of the brain, not directly related to the injury. And this is just quantification of the signal. And what is really important, we can also follow the same live mouse after stroke in 3 dimensions, and we can see this is the signal for refractory bulb, this is ischemic lesion and associated areas. How does it work in chronic? So here we see the first paradigm. We see event, stroke, or traumatic brain injury, and we see that there is a silent phase of inflammation and pathological events cascade lasting for several months. Now it's question, can we visualize silent pre-symptomatic disease in chronic models? The models that we're also using are models of amyotrophic lateral sclerosis or Lou Gehrig disease, and models of frontotemporal dementia and Alzheimer. So if you can see here again, this is the, for example. So this is the same animal that we started to image very, very early. So this is the SOD1 mutant mice. And in this mouse model, this is the model for amyotrophic lateral sclerosis. At this time, it's about 90-95 days, these mice start to develop hind limb paralysis. They start to be paralyzed, and they eventually die from a paralysis, like in a human disease. And these mice, if you look them in the cage, you wouldn't see the difference between healthy animals and the mice that already have some signals here. And what is really interesting, that we were measuring this biophotonic output, this is a big increase of the neuronal stress signal, and we know that the disease onset is just here. So we know that even before, if you look at in a 3 dimension, what we can see, this is the mouse that is completely healthy, looking by eye. So if you put the healthy mouse and this mouse, you wouldn't see any difference. But we know using this technology and having sensitive genetic markers, we know this mice already having a strong pathology. If we summarize, you can see in this mouse model, the disease is starting here, but you can see that the pathological signal from the brain and from the spinal cord are starting much earlier. Even in Alzheimer's, so this is the transgenic Alzheimer mouse. We cross with our genetic models. So in this model, there is an onset of cognitive deficit about 7 to 8 months, and you see even in the 5 months, at 5 months, we can see the pathological signal in the brain that involve in the time. Now question is, we know there are signals after injuries, the long-lasting pathological signals, and we also know that there are pathological signals starting way before chronic disease onset. The question is, can we use and map these signals and create disease-specific biophotonic profiles? And this is just to show you that for each of disease now we created our biophotonic profile. This is for ALS, this is for Alzheimer, this is for frontotemporal dementia ALS model. And why we are doing that? Because we know that knowing these symptoms, we know they are correlated with different behavioral deficits, we know what is pathology behind it. Now we can use the signature biophotonic profiles to test drugs, to do in vivo pharmacology, and to better understand from the live animal in real time what's going on in the brain. We can follow behavior together with the therapy, and we can see when they stop therapy, the behavioral defects are going worse, and this is completely correlated with the biophotonic signal, suggesting that this is really that this biophotonic and bioluminescence imaging approach represent a powerful tool to analyze pathological events and the therapeutic effects in real time, and it can be used in acute brain injuries such as stroke, as well as in the chronic neurological disorders. Here I will stop, and one more, next slide. Good. I will stop and put the next slide.